I am Peter ten Dijke, and I'm excited to share with you a recent publication in EMBO journal, which is entitled LncRNA LITATS1 suppresses TGF-β-induced EMT and cancer cell plasticity by potentiating TGF-β type 1 receptor degradation. This study was spearheaded by a talented PhD student, Chuannan in my lab at the Leiden University Medical Center and Oncode Institute. Epithelial-mesenchymal transition, in short EMT, is a key process for cancer cells to gain highly migratory and invasive properties. TGF-β is a main driver of EMT. In our study, we set out to investigate the role of long non-coding RNAs in TGF-β-induced EMT. For this, we performed transcriptional profiling to identify LncRNAs that are potently induced by TGF-β. We conducted the profiling in normal breast cells and breast cancer cell lines that respond to TGF-β-induced EMT. We reasoned that TGF-β-induced LncRNAs may also have a high chance of being functionally involved in EMT. Knockdown of LncRNAs revealed two classes that affected EMT differently. One set of which the knockdown promoted EMT, and another set of which the knockdown inhibited EMT. We focused in this study on the particular LncRNA of which the knockdown converted epithelial cells into mesenchymal cells and that enhanced the TGF-β-induced EMT. We termed this LncRNA LITATS1, which stands for LncRNA induced by TGF-β and antagonizes TGF-β signaling 1. Analysis of clinical datasets showed that LITATS1 expression correlates with better prognosis in breast and non-small cell lung cancer patients. Moreover, we found that in clinical samples, LITATS1 is higher expressed in normal lung tissues than lung cancer tissues. To elucidate the mechanism of action of LITATS1, we examined which pathways are changed upon depletion of LITATS1 using RNA-seq. Gene set enrichment analysis revealed that depletion of LITATS1 strongly positively correlated with the TGF-β gene response signature. TGF-β signals via the TGF-β type 2 and type 1 receptor complexes at the cell surface. The activated TGF-β type 1 receptor phosphorylates intracellular effectors SMAD2 and SMAD3. A complex of the inhibitory SMAD7 and the Smurf E3 ubiquitin ligase mediates the TGF-β type 1 receptor ubiquitination. This triggers its proteasomal and lysosomal degradation. We found that LITATS1 potently inhibits TGF-β-induced SMAD2 phosphorylation. Next, we examined the ability of LITATS1 to interact with the TGF-β type 1, type 2 receptors, SMAD proteins, and Smurf E3 ligases. To our great surprise, we found that LITATS1 interacts directly with the TGF-β type 1 receptor and Smurf2. LITATS1 promoted the interaction of the TGF-β type 1 receptor and Smurf2 and potentiated TGF-β type 1 receptor ubiquitination and degradation. In conclusion, we identified a LncRNA that serves to maintain epithelial characteristics of normal epithelial cells and that antagonizes TGF-β-induced EMT in cancer cells.